Okay, so enough housekeeping, on with the science. So very pleased to welcome Ausra Domanska from Sarah Butcher's group in Helsinki, and very much looking forward to a talk on the picornavirus. So Ausra, the floor is yours.
Thank you very much. I'm Ausra Domanska, and I will talk today about the coronavirus that can cause neonatal sepsis. So first, I will briefly introduce the coronaviruses. What are they? I will tell why it is important to study them. Then I will show you data on our virus reconstruction with icosahedral symmetry imposed, so to resolve capsid proteins. Then I will talk about asymmetric virus reconstruction, which allowed us to look at the RNA capsid interactions. And finally, I will present our latest data on reconstruction of virus in complex with Fabs from neutralizing antibody. So picornaviruses are small icosahedral viruses. They are non-envelope viruses, and they have single-stranded RNA genome. And this picture shows you phylogenetic tree from family Picornaviridae. This is a large family. It contains currently 80 species recognized, which are grouped in 35 genera. I would like to introduce, to say something about enteroviruses. They comprise many human pathogens, such as poliovirus, rhinoviruses, EV71, and so on. However, I will talk about pericoviruses, which are shown with this little arrow. This is small genus. It contains only 2 species, Pericovirus A, which includes human pathogens, and Pericovirus B, which includes Lungan virus, which infects rodents. Very briefly about the Pericovirus genome organization. So the genome is single-stranded RNA, about 7 kbs long. It encodes a single polyprotein. Coding region is flanked by UTRs. It has internal ribosome entry site, which allows polyprotein to be produced. Then polyprotein is cleaved into smaller fragments by 3C, which is a protease, viral protease. And then it's cleaved in even smaller fragments, smaller proteins. And I want to say that these are structural proteins, VP0, VP3, and VP1. And I want to point out here that in pericoviruses, VP0 is not cleaved in VP2 and VP4, which is seen in many other picornaviruses. So human pericoviruses, first human pericoviruses, 1 and 2, were isolated long time ago. However, the third one was isolated only in 2004. And this is explained by the fact that pericoviruses cause similar symptoms to those of enteroviruses. However, they do not grow on the cell cultures that were routinely used to isolate enteroviruses in diagnostic laboratories. And only when PCR-based methods were introduced to detect these, the other pericoviruses were detected. And now we have 17 genotypes. Pericoviruses mainly cause mild symptoms, gastroenteritis or respiratory infections. However, sometimes they are associated with severe diseases, especially in very young children, in newborns. And this is mostly associated with HPV3, the human pericovirus type 3. And there are no antivirals and no vaccines available for this. The only option that sometimes clinicians use is intravenous administration of pooled immunoglobulin, which comes from multiple donors. And it has been shown that this is effective if there is immunoglobulin that recognizes pericovirus. So we concentrate our efforts on studying, currently we concentrate our efforts to study pericovirus neutralization using human monoclonal antibodies. And the ultimate goal of this project is to find out some candidate antivirals. So we use structural methods to study our virus. So this is an example image taken by Titan Cryos Falcon 2 in Niesen in Netherlands. And these images were used for icosahedral virus reconstruction. I know that this is boring and you maybe all know about it, but I will briefly tell what is icosahedron. So it has 6 5-fold symmetry axes, which are marked with pentagon. It has 10 3-fold axes marked with triangle, and 15 2-fold axes marked with ellipse. And if we would imagine that this triangle is composed of 3 subunits, we would get this picture, which is basically picornavirus. So picornavirus asymmetric unit is composed of 3 major proteins, VP0 or VP2, VP1 and VP3, and VP1 occupies 5-fold axis, VP3 and VP0 occupies 3-fold axis, and VP0 or VP2 2-fold axis. So the icosahedral symmetry imposed during the reconstruction provides huge averaging power, which allows to reach atomic resolution or near atomic resolution from low number of particles, which is like less than 10,000. And I will talk about it just in a minute, how we achieve that. However, the data that are not following the same symmetry are severely reduced. And the most obvious object in viruses which does not follow icosahedral symmetry is genome, because it's like singularity. It's only one in the capsid. So, and I will show later how we dealt with this problem. So this shows a workflow for icosahedral virus reconstruction. So as I have mentioned, images were taken with Titan Cryos Falcon 2 camera, about 2,500 images. So as they were movies, 7 frames per movie, we used motion core to correct for beam-induced motion. We used CTF-3 for constant transfer function estimation. And then we picked particles using ETAN, about 55,000 particles. We used EMAN to inspect the particles, and we were left with about 40,000 particle set. First, we used Auto 3DM for our 3D reconstruction. This is program written by basically Baker's lab for reconstruction of icosahedral viruses. It allows to calculate random models from small particle number, like 150 particles. And then this model is used for as a reference model for final reconstruction. And with Auto 3DM, we got 5.7 angstrom resolution, which we thought that it's not really great. And then we used Relion. Again, we used the full data set. It gave quite low resolution, but after 3D classification, we got much smaller particle subset, which gave higher resolution after movie refinement. And I think that compared Auto 3DM and Relion, the classification and movie refinement, this is what brought improvement to the resolution. So and here I show you the particle. This is a typical picornavirus particle, which has 5-fold axis in here. And you can see that at the 5-fold axis, there is a channel. And it has also canyon, which is around 5-fold axis. canyon in picornaviruses is the place where usually receptor binds or antibodies can bind also there. In Pericoviruses, this canyon is very wide and shallow, and in other picornaviruses, it's more deep. So this makes the particle very smooth and without big bumps, and it makes it difficult for program to assign orientations. Yeah. So we used this map to build model, and we started with Icosahedral homology modeling for individual proteins using available structures from PDB. Either most of those were from crystal structures. So then these rigid these homology models were fitted into a map using Chimera. Then we cut out the asymmetric unit using zoning in Chimera, and did flexible fitting using iModFit and FlexEM, as just Maya described this morning. And then COOT for manual refinement, and then Phoenix for global refining. And we did it a few times just to reach some best outcome. And this shows you the asymmetric unit of picornavirus. So this is VP1, and this is the 5-fold axis, VP3 and VP0 at 3-fold axis, and here is 2-fold axis. And what is interesting in this, does not move for some reason. So we have a very long tail, which is here in protein VP0. And if you remember, in other picornaviruses, VP0 is cleaved to VP2 and VP4 during the particle maturation. And this long tail corresponds to VP4 in other picornaviruses. So, but in here it is not cleaved, and it was interesting to see where it, so compared to other picornaviruses, so this is unusual where this N-terminus is located. So these are VP0 proteins HPV3 from our structure, from hepatitis A virus, and from EV71 in its cleaved form. This is VP2 and VP4. So in enterovirus, this VP4 basically ends up in opposite position compared to this. And the structures kind of aligns quite well in this core region. So in context of the particle, this long tail travels quite long paths. So here are 4 pentamers shown from the inside of the particle. And this is VP0, and this is how it travels all the way and meets another N-terminus from another VP0. So we think that it maybe stabilizes capsid via these interpentamer interactions. So for comparison, there is asymmetric unit of EV71, and in black is shown VP4, which basically does not really go beyond its asymmetric unit. So let's look inside the virus capsid. So here I show 4 structures for Pericoviruses that are available now. So this is HPV1 early structure with 8.5 angstrom resolution. This is HPV1 X-ray structure from Plevka's lab. This is structure that I just showed you. And this is Ljungan virus structure. I think it's done in Sweden. So the lower panel shows on the right, no, on the left, it shows thick slice thick central slice, and on the left it shows, no, on the right, it shows rear half of the capsid. So they are colored, radially colored, and blue corresponds to the capsid density, and the pink for the RNA, for the genome. So, what we can see from here, that in the high resolution structure, we have very low density for the RNA, which basically suggests that there are very few identical nucleotides that are occupying these sites. And in lower densities, we see much more. In the lower resolution, we see much more density, which basically indicates that it might be some secondary structure that occupies this density. And the secondary structure might be very similar in all these cases. So but as these are icosahedral reconstructions, all of them, so how about the occupancy of these sites by the RNA? So for this, we used symmetry relaxed reconstruction. And we used the same data set that we used for icosahedral reconstruction. We just relaxed symmetry to C1, and then we got quite low resolution. And then we included all particles just to have a little bit better resolution. And then we got 10 angstrom resolution structure. And here is central section for this structure. And in comparison, this is central section for icosahedral symmetric reconstruction. And we can see very clearly how icosahedral symmetry can work with the same number of particles. So here we see the density for the RNA, and we still see it also in asymmetric reconstruction. So here I show you the density inside when the capsid density was removed. And what we can see in here, it will move or not. So that these densities are not the same, so the occupancy basically differs. So in these places you can see much more density than, for example, in these densities. So in pink it is shown RNA from the virus untranslated non-coding region. This is 13 nucleotides RNA, which was proposed to be highly structured, and it was modeled in RNA fold and RNA composer and was fitted in here. And in some those bigger densities, it fits pretty well, which kind of indicates that most likely something like stem loop structure could occupy these densities. So now I would like to introduce to another study, which in part was also done in our lab by Shabi Shakil, who was postdoc in the lab. So the goal of this study was to identify RNA sequence from the virus which interacts with the capsid proteins. So they used SELEX, which stands for Systematic Evolution of Ligands by Exponential Enrichment. And they used pentamers, which derived from the virus, disrupted virus, and RNA library, which was composed of 40 nucleotide-long RNA fragments, 10 to the power of 15 different fragments. And then after the SELEX, they identified matches or fits, and this is genome position, and these peaks show where they got matches. So basically, they got matches throughout the whole genome, but with different goodness, how to say, with different fit. And when they tried to do consensus of this, they found very short sequence, which is basically GUU, and this middle nucleotide even can be replaced with any other nucleotide. So, however, they noticed also that all these regions can fold into a stem loop structure, so that this motif, GUU, is presented on the loop. And this arrow shows you the first G here. So when this loop was taken, or basically not loop, or this GUU motif was taken and fitted into the density of HP1 crystal structure that I just showed, which had very low RNA density, so it fitted very nicely. So when you place A instead of G, the fit is not so nice, and this is difference map which shows these kind of regions which are not occupied. And they also show very nicely that they make contacts with proteins. So this is capsid tyrosine and threonine, which make kind of stabilize the capsid. So could it be possible that those kind of interaction sites could be possible targets for drug development? So this we don't know yet. It remains to be seen, but this could be nice drug target, we think. So speaking about drugs, many picornaviruses contain so-called hydrophobic pocket, which is located in VP1. VP1 is here, about this place. And here I show in red, it is VP1 from Pericovirus, which is superimposed on VP1 from enterovirus 6 to 8. And in gray, it's shown lipid factor, which usually occupies this hydrophobic pocket in picornaviruses. However, in Pericovirus, this hydrophobic pocket is blocked by the bulk amino acid chains, and basically there is no hydrophobic pocket as such, and lipid factor is not present there. And there are drugs that were developed against enteroviruses mostly, which could expel this lipid factor from here and occupy this place, and then they stabilize the capsid, and capsid cannot disassemble, and infection is prevented. So, but these drugs were shown in clinic earlier that they never worked with Pericoviruses. So, and this is probably the reason why. So now we were thinking about monoclonal antibodies. So maybe neutralizing antibodies could be possible therapy. And monoclonal antibodies were produced by AIM Therapeutics, by Tim Beaumont in the Netherlands. And here I show cryo-EM image of virus, human Pericovirus, in complex with FABs from neutralizing monoclonal antibody. So this is a picture taken with TechNight 20. So I used these particles from these pictures to make low resolution structure, which was about 10 angstroms, and I wrote application for INEX for access to high resolution microscopes, and I got access. I collected data with Titan Cryos in SciTech in Brno with Falcon 2 camera, about 6,000 images, and I did motion correction was done for movie alignment, with GCTF for CTF estimation, and then I used Relion 2 for all the processing stuff. So I picked particles. I got quite big data set. So after the declassification, about 180,000 particles. But after 3D classification, they were reduced about 32,000 particles. And auto 3D refinement gave me 3 angstrom resolution, which I am pretty happy about it. And well, I used structure from auto 3DM as the reference map, which came from Techni 20. So the nice thing was that motion core and GCTF were done on fly when data were collected, and we could already assess that data are good and we can continue collection. So, and here I show you the reconstruction, which is radially colored isosurface representation, and in orange you can see Fab molecules. So this part, which is most distant part, is not resolved very well because it is very flexible. This I could see from 3D classifications. However, the rest looks pretty nicely, and it shows that Fab occupies space around 5-fold axis, and it basically sits somehow between 2 asymmetric units, somewhere here. So I placed now by rigid fitting in Chimera the structure that I have from previous reconstruction. It is not so nicely fitting there, so I will have to do refinement, which I haven't done yet, so I can't really say what are interactions between molecular interactions between antibody and the capsid, which is basically my goal with this project. But I am kind of confident that I will be able to build a reliable model because the local resolution shows quite good. So the capsid region is quite well resolved, and it shows that the most distant part of Fabs is not very well presented. And this is the example how it looks like. So as a take-home message, or what we learned from these 2 reconstructions, so it looks like that addition of Fabs to the virus particle helps to assign particle orientations. So it might be, that's why I got quite good resolution. I haven't mentioned, but I used ultra-thin layer carbon grids, which might help for CTF estimation. It might be pretty correct. And then large data set allows to choose very homogeneous subset of particles, which also makes good for the resolution. So in terms of biological conclusions, so Pericovirus genome is highly ordered. So VP1, VP0 N terminus from Pericoviruses is most likely implicated in capsid stability. Hydrophobic pocket in Pericoviruses is blocked by bulk chains. So we have to think about some other therapies for these viruses. So this work was mostly done by Shabi Shakil, who was a PhD student and then postdoc in Sarah's lab. In Nissen, we collected data for the first at AIM. Tim Beaumont, he produced antibody, and recent data were collected at SciTech, and I'm grateful for Yuri about good assistance. And thank you for your attention.
Thank you very much. Questions? Pete?
So biological questions. So is it known which receptor is used by this virus to explain why newborns are so sensitive?
So no, we don't know the receptor for this particular isolate. We do know that HPV1 and some others, they use integrins, which bind to RGD motif, which is present in those isolates, but it's not present in HPV3, and it doesn't bind to integrins, and we don't know the receptor. And why newborns? It's hard to say. But in newborns, the blood-brain barrier is not really formed yet. And yeah, no, we don't know why, why it is neurotropic.
So I was wondering, this GXU motif that would occur randomly almost every 16 base pairs, presumably. So how sure are you of this, and have you done any biochemical or mutational validation?
Yeah, so some of these motifs were disrupted. I think the disruption was like loop, stem loop structure was disrupted, and virus production got reduced by 6 logs. So it looks like they are functional, at least some of them, not all of them.
Okay, if there are no more questions, let's thank Ausra once again.